Okay, well, welcome everybody to the C-DEBI Network Speaker Series. Today our speaker is Jessica Labonté. Before I do the more formal introduction of Jessica, let me just remind everybody that we have one final speaker for this series of 2019, and that is going to be James Bradley, who's now at Queen Mary University in London, and that'll be on November 7th. At the same time, and the title of that talk is The Power of Microbial Life in Marine Sediments. So hopefully you'll be able to join us for that talk as well. So today our speaker is Jessica Labonté from Texas A&M University in Galveston, where she's been an assistant professor since 2016. Jessica came to C-DEBI from a far-off foreign land, namely Canada, where she got a PhD from the University of British Columbia, working with marine viruses. She then did two postdocs, both at Bigelow, the first one working with Ramuna Stepanauskas, evaluating phage and single-cell data, and then continued at Bigelow with Beth Orcutt, working on extracting cells from low biomass deep biosphere samples. Then in 2017, she received a C-DEBI grant and will be presenting some of that work, I believe, today. And the talk title is You Are What You Eat, a geochemical and microbial study of a 3,000-year-old stratigraphic sediment succession. And just real briefly, when Jessica's finished and you have a question, the best way to do the Q&A is in the chat box down at the bottom of your screen. And with that, Jessica, I'll turn it over to you.
Thank you. Okay, I'm gonna make sure this is working. All right. So Jan said that I got a C-DEBI grant in 2017. This was like my first grant right when I started as an assistant professor. And I've had, so do you see all this camera too? Can you move? So you get this to see the screen. So I'm going to show you some of the results that we've gotten from this. I will show you a picture of her, but all this work was done by my graduate student, Catherine Risley, and she's gonna come here for the questions at the end to answer some of your questions. So we know that microbes dominate in subsurface environments, but they dominate pretty much any environment. They are present everywhere we are. As a microbiologist who studies subsurface samples, I am obsessed with microbes. I know they are everywhere. They're abundant, and contamination is it's a recurring problem. But in marine system, there's like 10 to the 29 microbes, but that's about as much as there are in the subsurface, and then in the sediment there's even about that same amount. So really a big amount of the microbial biomass is found in the sediment, and that's also true of the continental subsurface. And since sediment cover more than 70% of our planet, it's important to understand how they function and how they are impacting our environment. I really like this map that shows that there can be up to 10 kilometers thick of marine sediment in some of the regions of the world. And when we're thinking in terms of, so we know they're abundant in terms of biomass, bacteria, archaea, and viruses, they make about 77 gigatons of carbon on Earth. And obviously plants make up most, but that's because trees are much bigger than microbes. But in terms of abundance, microbes are still more abundant. And that out of these 77 gigatons, about 70 gigatons of biomass is found in the subsurface. And the majority of them are from bacteria and archaea. As we can see on the right panel, that in subsurface, which is the black, that's where most of the bacteria and archaea that we find on Earth are found, in the deep subsurface and marine environments. And because they're so abundant, they also play many important roles, whether it's in the carbon cycle, the nitrogen cycle, sulfur cycle, and all of this is really impacting the environment as a whole. So when we think of what's going on in the sediment, well, bacteria and archaea, they will respire, produce CO2, produce methane. They may eat that CO2, eat that methane, they will remineralize. And that's really interplaying with everything that is happening even at our scale on Earth in all these global systems. And we also know that they're very diverse in those subsurface communities. And I like this study because you think of the magnitude of the sediment. I told you in some places there's up to 10 kilometers of sediment. And here they drill down to 2.5 kilometers down and could still see microbial life. But we also see that there's still a stratification, that the microbes that are found at the surface are different than the ones that are found deeper. Some of these families are present all around, like the actinobacteria, but chloroflexi tend to dominate in environments that are above 400, up to 400 meters below seafloor. So we know that there's like a wide microbial community, and so now the question is, well, what are the drivers of microbial communities? And what drives microbial community and diversity? Usually we have to think about nutrient availability. And I'm just going to give you a debrief on redox, but basically microbes, they need to make their energy from these oxidation reduction reactions, and they will use a compound that is, and they use that to make their energy, or ATP. So you're going to have one compound that is going to donate electron and one that is going to accept electron. And usually the one here at the top, they are electron donors. The one at the bottom are electron acceptors. And the further the electron is moving down this redox tower, the more energy is made. So when we're thinking about organic carbon, usually they're at the top, they're good electron donors. At the bottom here, well, oxygen is a very good electron acceptor, but in our system or in many sediment, there's no oxygen available, so they have to use other electron acceptors. So what's happening with this dynamic between the nutrient and the electron donors, electron acceptors? So when we're thinking about the deep ocean, we're thinking of slower sedimentation rates, and usually these environments will be oxic, and there will be fewer cells because there's just fewer cells that are like less material that is piling up on the sediment. And because you don't have as much sedimentation, you don't have as many organic electron donors, basically. So your organic matter is a limiting source. But if you have an environment that have a high sedimentation rate, you're getting a whole lot of electron donors, but there's not enough electron acceptors. And that's why even if you're in sediment that have high sedimentation rate versus low sedimentation rate, we're still going to see pretty slow growth rate as a whole. And I'm going to tell you about the story that started it all for me, because that's when I was working with Beth Orcutt when she had samples from the Juan de Fuca Ridge from IODP Expedition 357. And we wanted to look at the microbial community, so look at 16S ribosomal RNA from sediment at the Juan de Fuca. And the idea was that we knew that nutrient availability is going to drive the microbial communities and be a driver. But the idea was, what if we sample closer or further away from the outcrop? So water goes in, it's changing its chemistry, water goes out. Technically, since the chemistry of the fluid is going to be different, there's going to be differences between the microbial communities. It's not exactly what we found. So we found that mainly the differences were based on depth, and mainly because it was like clay versus more the basalt and the sediment were completely different. But if it was a clay, it was different than if it was more a rock type. So we that made me think like, oh, well, if it's a clay, it's probably because whatever was there at the time of sedimentation was something that would impact the microbes that would be living in that layer, because those microbes would be limited by the amount of nutrient and the organic matter that they got at the time of deposition. And that's really what sparked the idea of the project that I'm going to tell you about. Another driver of microbial diversity, people who know me, they know I love my viruses. It's viral activity. So when we look at deep sea sediment, we can see that there are viruses like throughout the sediment column, and that in general, we see that they are more abundant when there is more organic carbon. So here we have the organic carbon in green and the viruses in red. And usually you have the same trend for each of these holes. These are from organic rich mud in the Baltic Sea from IODP expedition 347, and where you always have that same trend. And another thing that we're looking at is the virus to prokaryote ratio. How many microbial cells do you have for each virus? And that's a trend that has been looked at in different type of sediment, coastal sediment or other sediment that are much more nutrient limited, like in the South Pacific Gyre. And what was noticed is that in general, as you go below, like further down below the seafloor, the ratio of viruses to host is increasing. So, and there are a few scenarios to explain why would there be more viruses compared to the number of cells as we go deeper. First of all, it could be that viruses are just more preserved in these environments, but it could also be just if we're thinking again at those oxic sediment where there's not a lot of sedimentation, there's not a lot of cells to begin with. If one virus is successful in lysing a cell, well, the time it's going to take to diffuse to go infect another cell is much smaller. So you're going to have more viruses compared to the number of cells, just for the simple reason that they don't necessarily get to the next cell. While if you're in an environment where there's a lot of cells, because there's a very high sedimentation rate, where you're going to have a lot of viruses, but these viruses are also like there to lyse the cells, providing nutrient for the other cells. And viruses can also be themselves nutrients. So as you go deeper and deeper, you're going to have less, like as the amount of cells go down, you're also going to have fewer viral particles, and the ratio is not going to be as high. And that's what we also observe in our system, that is an anoxic sediment with a very high sedimentation rate. And just here to review for the ones who are not as familiar with viruses, there's two different life cycles for viruses. They can either enter the cell, take control over the cell, replicate, and then it ends in the cell bursting, releasing viruses. And that's what usually happens here in those sediments. You see the cell will burst, releasing viruses. Viruses can then diffuse, potentially finding another host. Some of them can also integrate inside the genome of their host. They're called lysogenic. It's the lysogenic cycle, and these viruses are called temperate. And they will actually integrate inside the genome, and that can be a driver of evolution. They can carry genes from one host to another, and so forth. So that brings me to my study site, which is the Blackwood Sinkhole. It's located in the Bahamas. So our site is not in the deep sea like most of the IODP expedition. We wanted to see if this site, which has a very high sedimentation rate, but it's also anoxic, just as it would be in the deep sea, It's very rich in organic carbon, and it is groundwater fed. So the interesting thing is that this sinkhole, so here we have the Blackwood Sinkhole, there's going to be a lot of terrestrial input if there is flood water or rain. But it is, because it's groundwater fed, at the bottom, it's all salt water. So the sediments are actually in salt water, because it's about 220 meters from the beach, and the water comes from the ocean through the bottom. So our questions were, well, are the subsea floor prokaryotic communities comparable to the ones that are inhabiting this anoxic oceanic basin? So we go on these expedition with IUDP. They cost a lot of money. Could we use results from a sinkhole to potentially plan better our expeditions and design our experiments? Is it similar enough so we could do that? Our second question was, well, are there differences in organic carbon rich or organic carbon poor sedimentary layers? It has a very fast sedimentation rate, so you get to see many of these layers. And so are there differences if 1 year there was fewer carbon versus more carbon? And the last question was, well, what is the extent of nutrient regeneration in subsea floor sediments by viruses? So what is the extent of viral lysis? Is it working like I showed you, where they're going to burst many cells and releasing the nutrients, or is there not much going on? And that really is impacted by microbial activity. So the reason why I decided to study this site was, so my collaborator, Pete Van Hingstum, he's here also at Texas A&M Galveston in the Marine Sciences Department. And he is a paleoclimatologist. So he had multiple sediment cores where he reconstructed the climate. So he did radiocarbon dating. So we had, for each of the cores, you can exactly know how old it is by using the radiocarbon dating. And you can look at the grain size, for if it's fine versus coarse, and the carbon to nitrogen ratio. So if the carbon to nitrogen ratio is high, means you have a lot of leaves and wood, and usually it's also going to be associated with a coarse grain. But if the grain size is very fine and you have a low carbon to nitrogen ratio, it means you probably have sedimentation that is more associated with just normal algae or particulate matter that was in the water column that is sedimenting. So doing that, he was able to look at the climate, recreate the climate over the last 3,000 years, and see when there were periods of high hurricane seasons versus lower hurricane seasons. Another thing that was done on this site was to find the source of organic matter. So I told you, if there is a storm, there's going to be a lot of water, a lot of rainwater, a lot of wind. So there should be more pieces of wood. There should be lots of leaves. And look at the Blackwood Sinkhole. So here we have two sites. There's a green one and a blue one. So our course that we took for the microbial activity was more, it was taken in the middle, right next to this BLWD C3 core. So the blue one. So is the organic matter more similar to what it would be if it's coming from terrestrial, from the ocean, or from wetland? And we can see that in general, because we're looking at the middle, when the water is seeping through underneath, it is more similar to wetland and coastal environments in general when we look at the carbon. And in terms of nitrogen, a lot of the nitrogen is similar to what would be coming from wetlands. So that's one difference, that if we decide to use sinkholes as some sort of an analog to to the deep sea, that's a big difference, because sedimentation, the particulate matter is not just coming from sedimentation from the water column. And finally, another thing that they did on this specific site was to look at the vegetation history over the last 3,000 years. So in the 90 centimeters of cores, it can go back to about 3,000 years. So that's about the age of the sinkhole. And we can see that there was some sort of a transition of the vegetation on the island. And that means that that it impacted whatever organic carbon was imported into the sediment. And we can see in yellow here, there are 3 yellow boxes, and these are regions wherein there was increased primary productivity. So and that is reflected when we look at more the foraminifera, which is another study that they have been doing, but I don't have the results for this one. But that's something we can also do, look at the evolution, and that's also showing us if there are like, when did the water started seeping through, and that was about 1,500 years ago. At first, it was just fresh water, and starting about 1,500 years ago, salt water started seeping through. And these regions of like primary productivity, high primary productivity, I listed them here. So, and I'm going to show them to you every time on the core, so we can to see the differences. But there's like 1,500 to 900 years ago, 700 to 500 years ago, and 300 to 100 years ago. So what did we do? We took a fresh core, and that was collected in July 2018. We did not use the old cores that were stored here in the fridge. We went there, collected a brand new one right next to that C3 site where most of the studies had been done. And here we have a picture of our core. We have a radiograph, a picture of our core. And what we did is we divided it based on the stratigraphy. We divided it into 24 distinctive layers. And each of these layers had between 21 and 90 grams. And that's going to play in when we do our extractions, because we needed 5 grams for virus-induced microbial mortality experiments I'm going to tell you about, 0.5 gram for total DNA extraction to be able to do the 16S ribosomal RNA gene amplification, 10 grams for total DNA extraction for metagenomics, and 40 to 50 grams for total viral DNA extraction. Viruses, they're very small, their genomes are very small, they're very hard to get from out of the sediment. Therefore, we need way more material to be able to have enough to do genome sequencing. We also took some samples to do radiocarbon dating. We don't have the results yet, but that will really help us put our core into the same context as the other ones, so we can really compare the carbon to nitrogen ratios, and which we also sent some samples for. For each of the layer, we did carbon to nitrogen ratio, and we took some samples also for the water, pore water geochemistry. So our first question, are the subsea floor prokaryotic communities comparable to the ones that are inhabiting anoxic oceanic basins? So here, for every time, I'm always going to show you the core with its radiography, the picture, and the 24 layers that are alternating green, gray. And in black here, these are the 3 times of primary productivity that were high primary productivity. And these are, at this point, because we don't have the radiocarbon data, they're not arbitrary, they're estimated. But then we did the 16S ribosomal RNA gene for all of these 24 layers. So what we can see from this is that in general, we can, the microbial communities are fairly similar to the ones that we would find in the upper sediment. We have a very high concentration of chloroflexi. We have a lot of archaea and a lot of proteobacteria. And one interesting thing from this is that whenever we have these zones of high primary productivity, there seems to be a spike of these epsilon proteobacteria that are now called campylobacteria. So maybe they are influenced by what they were offered to eat, which is whatever if there were more algae, then maybe it is a driver for these epsilon proteobacteria. We also did the carbon to nitrogen ratios to compare to what had been done before with the other two cores that were sampled, and the carbon to nitrogen ratio can tell us a little bit about what was there at the time of deposition. So if you have a high carbon to nitrogen ratio, it means that you have more leaves, you have more cellulose, and those are not carbon compounds that are necessarily very digestible by bacteria. While if you have a low carbon to nitrogen ratio, it's usually from algae sedimentation, which is an easier nutrient form for microbial activity. So we can see that there were some variations. I think I put, sorry, I thought I kept the legend, but in purple, it's the ratio, and in black, it's the total carbon. And so we can see that there was total carbon between 5% weight to about a little over 20, while the ratios is, some of them had very high ratio, and this one that has the highest ratio was actually, it looked like a pile of leaves that were piled up in the sediment layer. So it fits with this, that it's more leaves and cellulose. Some of them were like the sediment, they're anoxic, so it's almost like a way to preserve this organic matter. Some of them were really a texture of tree bark that had been there, and those can be associated with heavy rainstorms. And what if we compare this carbon to nitrogen ratio to the 16S that I showed you previously? There's not really a big trend between the carbon to nitrogen ratio and the microbial community, but there is a bigger trend when we look at the primary productivity regions of high primary productivity. As I mentioned to you, where we had these epsilon proteobacteria that were more prevalent. And we took 6 samples that we sent off for metagenomic sequencing and also for pore water geochemistry. And you may be wondering, why did we choose these specific 5 samples? Well, I told you we had between 21 and 90 grams of sediment for each of the layers. We went for the layers that were the thicker, that we had the most materials, so we were able to do metagenomics of both the bacteria and archaea and the viruses. But in the end, our idea of we had a wide range of carbon to nitrogen ratios, and that's lucky on our part. We got some of the highest and some of the lowest as well. So we were still able to answer our questions. And we also did some nutrient analysis of the pore water. So we did the nitrogen, phosphate, and silica. And here I just kept the 6 layers for the 16S ribosomal RNA, so it's easier to compare. And I put again the regions of primary productivity as well. Like if you notice, I always keep the same colored stars for each of the samples because they don't not all the layers were exactly the same thickness. So we see that at the beginning we had a spike in all of the nutrients, especially nitrogen and silica, and then it drops and it goes back to like once we reach the bottom of the core. And we see differences, and those are not necessarily related to the carbon to nitrogen ratio, nor the 16S, but they are more correlated with the metagenomic data, as I'm going to show you. So this part of the question, we're not done. We are still getting some of the data. So we're waiting on the radiocarbon dating that's going to be coming soon. So we can do like this, put the timeline on our core and be able to compare it with all the other analysis that were done on the cores that were nearby the one we have. And we also need to see if there's any correlations between the carbon to nitrogen ratios, the nutrients, and the microbial distribution. So our second question was, what are the differences in microbial communities in organic carbon rich versus organic carbon poor sedimentary layers? So to do that, we used metagenomics. And I'm not going to go into all the details of the softwares. If you have questions, you can come to me. But basically, we got about 30 million reads for each of the sample. We merged the reads, did some quality trimming, did de novo assembly, gene prediction, and functional taxonomy and assignment. And today I'm going to show you some of the results we got out of Megan, because it's easy to compare all the samples. I'm not going to show you the binning, but we did get quite a few genomes that we're analyzing right now, so we can put the metagenomic into a context of who is doing what. So how do the samples compare? Because we only had 6 out of our 24 samples. And they were chosen originally based on their carbon to nitrogen ratios. Well, it looks like the samples really follow this trend when we compare the metagenomic data. So we have this 72 with an extremely high carbon to nitrogen ratio is different than the other ones. And then it goes, mainly you have the top layers that are more similar, 6 and 24, and the bottom layers that are more similar, and that 35, which is pretty much in between, is in between those two groups of samples. And when we look at the microbial communities, so this is taking all the genes, all the proteins, and comparing them to GenBank, and then putting these GenBank results back into a taxonomic context. We can see that the main groups of bacteria, like they are like chloroflexi, like is quite dominating. Disulfo bacteria, the delta proteobacteria that are able to reduce sulfur, they're still dominating. So it is somewhat similar to the profiles we would get with the 16S ribosomal RNA. But what if we take this exact same data and just sort it to see, can we see differences between each of the layers? So from the left, now instead of going from up to down, we're going to go from the left to the right. We have our 6 samples from shallower to deeper. And then the bacteria were sorted, the taxon were sorted based on the Z-score. And it looks like we have a trend, and the Z-score is going to amplify the signal, what is enriched in layer at 6 centimeter versus what is enriched at 72 meters. And we see that there is a transition where we have our sulfate reducers in the shallower region and all the way to the deeper. But there is a transition from shallow to deep. There is, how does this translate in terms of metabolism? So we can also look at the seed assignments for each of these genes. And again, we see a trend where we have sulfur, phosphorus, photosynthesis that are more, and photosynthesis in this case would be the microbes that they don't do photosynthesis in the sediment, but it's the ones that sedimented. from this sulfur and phosphorus metabolism to a lot of carbohydrates, and then polyamines. And those fit actually our nutrient profiles, where we had a lot of nitrogen at the beginning, like in the first and the last sample, and that's what we also see with our nitrogen, and the polyamines as well. And the silica is associated with the algae that are actually sinking. So we see that photosynthesis, the silica was high at the beginning. So we can see a trend also between our like the metabolic pathways and the diversity that fits with the nutrient profiles. And we're still working on deciphering all of this, but it is encouraging to see that we kind of see trends. And that 68 centimeter, the one that was very leafy, that had a very off carbon to nitrogen ratio, is completely different than all the other ones in terms of the metabolism that are going on out there. And for this now, we're trying to identify who is doing what. And we got about 30 genomes that this is just one of the sample, just a few of the genomes, but we have multiple bins. We separated the reads into bins, and we're keeping the ones that have at least 90% completeness and not too much contamination from potentially other bins. And we got some gamma proteobacteria, some archaea as well. And now we're trying to see what are the role of these bacteria, putting them back into the context of the whole metagenomic communities, and also looking at the nutrients. And our last question was, what is the extent of nutrient regeneration in the subsea sediment done by viruses? And to do that, we did virus-induced microbial mortality experiments. So you take your sediment, you make a slurry, and you incubate it at the same conditions that it was in the environment for 0, and then take samples at 0, 1, 3, 6, 12, and 24 hours. And then for each of these samples, we separate the viruses from the sediment and the bacteria using sonication and sodium pyrophosphate. And we count the bacteria and the viruses. And then we can see if there was an increase or decrease in bacteria or viruses. And we use epifluorescence microscopy to count the cells. So again, I'm bringing these two back, where we have our carbon to nitrogen ratio and our nutrient profile. And we have the bacteria and virus counts for each of these 6 samples. And what we see is that for the sample at 24 and 68, or is it 6? For these 2 samples that have the lowest nutrient concentrations, the viral activity was pretty much not very, the viruses were really not active. And that's an indication that their hosts were most likely not active either, which, and that fits with the idea that even though there may be quite a bit of carbon, they ran out of electron acceptors. And the ones that had the most viral activity were the ones where the nutrients were at the highest concentrations. So really the viral activity depends on their host, because a virus by itself cannot do anything. It needs a host, and then the host depend on if they can produce energy or not. So now what we're doing, so we also got viral metagenomic data, and we got plenty of viruses. We're just trying to analyze to figure out what those viruses are. We got archaea viruses, bacterial viruses, and trying to link them with their host using bioinformatics tools. And we're also working on trying to identify viral sequences, but in our bacterial DNA metagenomics. And that can help us seeing if there were viral infections, if they are more lytic viruses, or if there are prophages. And we did use some software and found some, but it takes a while to analyze all of this data, so I didn't have a nice story to tell you. But we do have both lysogenic viruses and lytic viruses in our bacterial metagenomic data. So in conclusion, to the question, like, are the subsea floor prokaryotic communities comparable to the ones inhabiting anoxic oceanic basins? To a certain degree, they are, because the microbes themselves, like, they share quite a bit of similarities. We have the same kind of dominance with chloroflexi, the proteobacteria, and upsilon proteobacteria. But what we have to be careful, if we want to use sinkholes to generate data, for preliminary data to do scientific ocean drilling, is that the nutrient sources are vastly different, and that's something to take into consideration if we want to plan further experiments. Were there differences between organic rich and organic carbon poor? And that's a tricky one, because even our most carbon poor are still not carbon poor, but we could still see differences, but they were not mainly based on the carbon. There seem to be differences more because of the ecosystem productivity, and also when we look at the other nutrient, the electron acceptors seem to definitely be the limiting factors. And now what would be interesting to do is correlate these differences in the diversity and genomic potential and see microbial activity. So we could look at enzymatic activity or RNA to see really how are these microbes really, how are they active in this system. And finally, what is the extent of nutrient regeneration by viruses? It seems that, again, viral activity is much higher when nutrients are available, because you need these electron acceptors to produce energy. So that seems to be the key as well. And now the type of questions, now that we have found prophages and like that we have viral sequences, we can see if viruses are drivers of evolution in this kind of system, and also try to see which members of the community are most impacted by viral lysis, because when we're going to compare our viral metagenomic data with our bacterial metagenomic data, we can try to link who's infecting who. And as I mentioned earlier, none of this would have been possible without the members of my lab, but especially Catherine Risley, who is graduating soon as a master's student. All of this is her work. And also Piet van Hengstum with the micropaleoceanography lab in marine sciences. Anita Malavaj really helped with the C/N ratio measurements and also funding. C-DEBI funded most of this. Catherine got a C grant, graduate student grant in aid to help with the chemistry analysis and getting data for the nutrients. Thank you. I'm ready to take questions. Okay, so did we infer viral activity solely from relative abundances? So we did, yes, we did do it from the activity, but from the incubation. So it's over time, but that's how we did it. We couldn't measure it on the site. Those cores were replicates of the ones that were collected by our collaborators. So yeah, and he actually took it right next to the core that we had previously, and that's why we decided to do the radiocarbon dating, so we can really compare exactly what layer, because the sedimentation rate is so high, it actually can add, like, quite a bit. Even if it's 5 years, it's gonna be off in the depth. And the last question is, if they were stored, do you think storage time and condition influenced our activity measurements? That's why we took a new core, because the old cores had been stored for over 5 years. So we didn't want to use that core, and it was not necessarily stored under the proper microbiology conditions as well. Next question, to what degree do you think that dead viruses are able to persist in sediments? That's a very good question. And I think they can persist for a long time until they are eaten, basically, because we can store viruses for very long times. Do you want to answer a question? No. Okay. Catherine is here to help me if I have problems. What species of mangrove were there? I think to this extent, because I'm not an expert in mangrove, I can only refer to you at this point on this where there's so there used to be palm trees, but no more. And right now, it says, yeah, I'm not a tree expert, so I would have to refer to you to this image. But if you have more questions, you can email me about that, and I can definitely give you the right answers. Stable nitrogen activity. Oh, so the more negative stable nitrogen activity we find with nutrient limitation trees can take nitrogen from the air in sulfate reducing systems. And that's a very good point, and I'm going to write it down, and we can definitely look at that because we can compare our metagenomic data. We have sulfate reducing microbes, and we could see if they are impacted by actually the nitrogen. Okay, so regarding the heat map, so this heat map was built at this point. It's not binned, it's really just, we took all the genes from every contigs and put them into BLAST and then in Megan. So it's not based on, the first hit that it's going to classify it as flavobacteria or Bacteroidetes. And now we're working with our bins to go and back into these assemblies and put them into the context of our metagenomic data. So we're sort of doing things in double with just single or binned. I hope it answers this question. So that leaf, so question from Stephanie Carr saying like if that leaf layer had different bacteria, and when we look at it, it doesn't look like the bacteria were that much different. But again, this is still at the phylum level, and the reason why I presented it at the phylum level today was because once we go down, it's so diverse that we cannot make a graph, but we are planning to go and look at order and class level to see at the fine grain, do we see differences among these layers? But for graphing purposes in the presentation, I did. Is there sulfate, sulfide geochemical profile to the core? There was not enough, was it? It was below detection.
That was the nitrate. It was below detection.
Okay. Nitrite. So the question is sulfate, sulfide profiles. I don't believe we have those.
No, we weren't able to get them.
We were not able to get them, but nitrite were below detection level for the whole core. And yes, myrnaj, please send me the article. Are there other questions? I can't even see my camera anymore. Somebody's coming.
All right. Well, thank you, Jessica, for the great talk.
Thank you.
Yeah, so please stay tuned for our next speaker series talk coming out in about a month with Dr. James Bradley. And for anyone who missed this talk, we'll be posting it to YouTube shortly. And thank you again.
Thank you all.